All right, so we are on the cusp of our next exam. I would imagine we'll have a few more people joining us as per the usual, but what we're going to be doing is finish introducing our different chapters that are going to be included in this exam and talk about anything else in terms of specifics, like the fact that this exam is going to be on campus this time. So that will certainly be something a little bit different. So we'll talk about the details of that as well. So with the on-campus meeting, that meeting will be occurring, remember, at 6 p.m. instead. So it's a little bit different than what we had seen previously. So this will be, rather than 8 p.m., it's going to be 6 p.m. 1 week from today, and it's going to be in Fountain Hall 217, so FH 217. Let me type that in. Exam 3 is in Fountain Hall 217 at 6 p.m. Also send an announcement or email through Desire to Learn. So that exam is going to be the time when you are going to need to shower to come to campus. You're going to need to bring a few things with you. You will need to bring a Scantron form, a Scantron 882 form, which is the rectangular blue-green colored form. It has 50 questions on the front, 50 questions on the back. So you're going to need one of those for your multiple choice section. The rest of it you'll be able to write on the exam itself, like the lab section and the short answer essay section. You'll also be wanting to bring a self-addressed stamped envelope. So the self-addressed stamped envelope needs to either be like a manila envelope, which is 9 by 12 or whatever, or it can be, gosh, is it called a legal size envelope? You know, the typical one that's 9 inches wide. Trying to figure out if it has a particular name. But, you know, the regular size envelope, you might receive a bill in or something like that. You could certainly bring a self-addressed stamped envelope that's one of those sizes that's 9.5 inches wide. So a self-addressed stamped envelope for the return of your Exam 3. Now, if you send a regular white envelope, you can put one stamp on it. If you send the 9 by 12 manila envelope, you'll need to put 2 stamps on that for the return of your exam and Scantron once I get done grading. Now, in that self-addressed stamped envelope, you can also put any proof of extra credit from extra credit that you have accumulated to date. So you can either turn it in now or you can turn it in on the day of the final in December. It's entirely up to you. But putting that proof of extra credit inside of your self-addressed stamped envelope is a good way to get it to me at this point. Okay, so in terms of what we have to expect in the up-and-coming exam, remember that this exam covers our chapters 9 through 12. So it's the muscle chapter, the control of body movement, that smaller chapter there, endocrine system, and then cardiovascular physiology. In terms of labs, we have complementary labs, all Physio-X exercises. We have a skeletal muscle physiology, Physio-X exercise, an endocrine system one, and then 2 cardiovascular associated ones. We were going to do exercise 5 and exercise 11. 5 is one of our cardiovascular physiology exercises, and then blood analysis is exercise 11. So we're going to do that one as well. So we'll have questions on those 4 Physio-X exercises. So what I wanted to do tonight is go through the remaining material that we haven't gone through yet, answer any questions from chapters you've gone through before, and then kind of move forward. I also wanted to propose, and I can send an email about it, that if you like, you know, we could try to do some sort of review session or something like that, or last-minute question and answer session on Tuesday of next week prior to that Thursday exam. So that's a possibility too. So we'll see how things go. We did have a quiz that was originally due yesterday on the first few chapters associated with this exam. As I sent in the email earlier today, most people took it on time, but there are a few of you that did not. So I opened it up again to be open through midnight tonight. So if you haven't gotten it done, make sure you do. Those that got it, having to get it done late, will have a couple point deduction on it, but that is much better than getting a 0 on it, to be sure. So make sure you get that quiz done if you have not. We'll also have an additional quiz, quiz 6, that will be available this weekend, and we'll run that one through Tuesday of next week. So we'll run that one through like Tuesday night, midnight of next week, and that way you can get your results back and be able to study Wednesday and then Thursday before that Thursday exam. So any questions about any of that or anything else that you have dire need to communicate? I'm pretty quiet out there so far. I assume you're hearing me because I can see my little speaker thing, my microphone bar moving. You guys there? Go ahead and give me a check mark yes or an X no. David's typing. He's going to let me know he's there. There we go. Okay. It's not just me. Just making sure. Oh, thank you very much. Nervous about this test. David, why are you nervous about this test? A lot of info to study. Do you feel it's a lot of info to study different from other exams? Last one we had the nervous system, which was pretty rough. Or it's a lot of detail. Just an overall feeling about it. Well, and cardio is big. We're going to go through that tonight. Well, I mean, I think to a certain extent it is a good thing to be nervous or at least concerned about it at this point, because given the fact that we still have a week to go, we've got time to put that concern to good use in terms of getting that studying done, feeling more comfortable with it, really nailing it down. So if it was the day before exam and you were feeling really nervous, then that would be a bit more of a concern. I think at this point we're going to be okay. So remind me, I believe last time we went through both the control of the muscular system, and then didn't we also go through endocrine system? Is that true? Right. Okay. So people made some flashcards for endocrine system, that type of thing. Just a suggestion. You certainly don't have to, but it's that way of getting little doses of the content rather than having to worry about all of that content at once. So if you haven't done it yet, I would consider doing it to be sure. So the next quiz will basically be over cardiovascular system because, as Dave mentioned, it looks pretty big. So we'll have that. Will be the one that will be due Tuesday night at midnight, next Tuesday. So let's take a look at that cardiovascular system and see if we can kind of get our heads around it and organize it in terms of our thoughts. So what we're going to find is it certainly is a big chapter. I think I referenced last time the fact that often in textbooks they will actually separate the cardiovascular system into 2 different chapters. Usually a smaller one that is blood, and then another one that is the cardiovascular part. Sometimes they'll separate it into 3 chapters: the cardio, the vascular, and then the blood. But regardless, it's the same information. It's just a question of whether you have a big picture and a number associated with the beginning of a chapter, or whether it's all together like it is in our book. But that, I think, is really kind of where you want to focus, though, your effort in looking at those 3 different components. So we want to look at and talk about the heart, we want to look at and talk about the vessels, and we also want to look at and talk about the blood. So let's take a look through and see what there is to see about this cardiovascular system. So it does a little introduction to the blood, but really most of the content for the blood is towards the end of the chapter, so we're going to wait on that. It does some introduction to the system overall, and then it gets into our blood vessels, as well as a little bit of what we would call hydrodynamics. So something that we're going to see here as we're looking at the system is, and let me get back to that original diagram, is that with the system, it's a circuit, right? So what we have is the pump in the center, the heart, and then we have 2 main circuits. We have the pulmonary circulation, which is going to and from the lungs, And then we have the systemic circulation, which is going to and from the rest of the body. Now technically there's a third circulation, which is called the coronary circulation or cardiac circulation, where the heart itself has arteries and veins going to it and from it that serve it as an organ and its metabolic needs. But mainly we look at these two large circulatory patterns. But with a loop like this, or even two loops that you see here, anything that occurs along this loop is going to affect other parts of the loop. So one of the things that we're going to see is anything that occurs with the heart, if we change the heart rate, if we change the strength of the contraction or something like that, that's going to affect what's going on out in the blood vessels. Likewise, whatever's going out in the blood vessels is going to affect the heart too. So the amount of blood that we return back to the heart in these different veins from the blood vessels is certainly going to affect the heart's performance. If there's any blockages in the arteries and veins, that's going to change the pressure and the flow and things like that out in this circuit. Even if we talk about changes in diameter of those blood vessels, we'll notice even in this course, if you will, course with an A in it as opposed to fine, even with this course diagram that we have here, we can see there's a change in terms of the diameter of these blood vessels. And as we get smaller and smaller, these blood vessels will conduct, an individual one will conduct less fluid in it, right? There's simply, it's a smaller tube, a smaller pipe, so it can't have as much volume going through it per unit time. That small diameter also increases the pressure leading up to it because there's that backup, if you will, and flow, everything trying to get through that small diameter pipe. So the diameter of the pipes that we have here has an effect on flow. So one of the ways that we can homeostatically maintain the cardiovascular system is not only by changes in the heart itself, but by active changes that occur out in the blood vessels too. And it's all hooked together. Now this is certainly a key point that you need to understand from the get-go, is that there's this rule that all arteries carry blood away from the heart, and all veins carry blood back to the heart. A lot of people get stuck on this thing, oh, oxygen in the oxygenated blood in the arteries and deoxygenated blood in the veins. And while that is true in that systemic circuit, it is not true in the pulmonary circuit, right? The rule is that arteries always go away from the heart, right? The A's match up. So here, for instance, if you see the pulmonary artery, we can see it going away from the heart, but we can also see that it carries deoxygenated blood. So it's going away from the heart, right? But that's deoxygenated blue blood. It's going to the lungs in order to pick up oxygen and get rid of carbon dioxide. And then, in the vein, we're going to see the blood returning to the heart. So we can see that that whole idea of a red artery and a blue vein does not hold up in all cases. Okay. So like I said, from time to time we will get into a little bit of the physics of the situation because we're talking about fluid dynamics, right? So, you know, one of the examples that sometimes I use when I'm teaching this to even like elementary school classes and stuff like that, as I talk about the differences that you would have in terms of like if you were trying to drink a drink with a regular straw or with a stir straw, right? The diameter of that tube affects the amount of flow that you can get through it. So that's going to come into effect here in the cardiovascular system. It's certainly also going to come into effect in the respiratory system, which is what we address right after exam 3. Because that also has a series of tubes as part of that system, but in that system, rather than conducting fluid, it's going to be conducting air. But same thing, bigger the tube, easier the flow, smaller the tube, the more difficult it is for the substance to get through it. Okay. So then, the chapter gets into the 3 basic parts. So we start out with heart, we go to blood vessels, and wrap up with blood. Now with the heart, we start out with some basic anatomy. We talk about the different chambers of the heart that you can see that most people know already, right? You've got atria up towards the top. You've got ventricles that are more inferior, and they are separated by different valves, right? Atrioventricular valves. There are also valves here which are those that keep the blood from flowing back into the ventricles from the attached arteries, either the pulmonary artery or the aorta that we see there. And so it's a very regulated system where on the right side of the heart we have that deoxygenated blood, and on the left side of the heart we have oxygenated blood. So we see different chambers. We also see a very important separation between deoxygenated and oxygenated blood between the left and the right side of the heart. Talk a bit about the layers and the valves and things like that. But then, just like usual, we kind of spiral our way down into the cell, right? Because in order to understand how the organ works, we got to get down to the tissue. In order to understand how the tissue works, we got to get down to the cell. Now, when we look at the layers of the heart, you'll find that most of the wall of the heart and the layers are made up of cardiac muscle tissue. Now, as we found out when we were doing skeletal muscle tissue, the main reason that muscle tissue is around is that it's there to contract, right? So it contracts when you stimulate it. But we're going to see some differences between what we learned in skeletal muscle and what you will hopefully then learn in cardiac muscle. So it's going to be very important for you to be able to compare and contrast these things. So, you know, you could have a question where it has you draw out the membrane potential change, you know, those graphs like the depolarization, the repolarization, the hyperpolarization, that type of thing, where you're learning about action potentials before in our other excitable tissues, whether it be skeletal muscle tissue or whether it be a neuron, right? Comparing what we learned before with what we would learn now in terms of cardiac muscle tissue. So that would be something good to understand. The other thing that we'll find out is that the cardiac muscle cells, we have two distinct populations of cardiac muscle cells. Now that's certainly a unique situation, right? Usually when we get down to the cellular level, like neurons in the brain or something like that, there's just one functional cell that provides that function to the organ. But here we're going to find that there are two different populations. One type of cell are called autorhythmic cells. And autorhythmic cells are going to be those that, as the name indicates, are going to automatically generate a rhythm. So these are going to be the localized control centers that generate the basic heart rhythm. So these are the managing cardiac muscle cells. We're also going to find, well, and looking at this figure here, you see all of this yellow that's in here, okay? This represents part of what's called the conduction system of the heart. And so the conduction system of the heart, much like if we're talking about like a symphony or something like that, right? You have the conductor, which directs everybody. Now there's only one conductor, there's very few conductors, and there's a whole lot of symphony members that are performing the action led by the conductor. Same thing with the heart. Those autorhythmic cells make up the conduction system of the heart. They're only 1% of the population of the cardiac muscle cells, these autorhythmic cells. The rest of the muscle cells are the ones that are acting upon that direction. And those are the contractile cells, which are also cardiac muscle cells. So there's another kind of important comparison that you can make when you look at, we talked about contractile cardiac muscle cells, which is what we were thinking at first. Well, what other type would there be? They're just cardiac muscle cells. They'd be like skeletal muscle cells, etc. So you'd want to compare the contractile cardiac muscle cells with like a neuron or a skeletal muscle cell. But you could also do a compare and contrast between the contractile cardiac muscle cells and the autorhythmic cardiac muscle cells. So when you look at this graph that you see up towards the top here, okay, this is for the contractile cells. This is for the other 99%, if you will, that are doing the contraction. You'll notice that we see quite a bit of a difference here. In the skeletal muscle cells and in the neurons, it was only a handful of milliseconds when that action occurred. Now here in the contractile cells, when we're looking at changes in membrane potential over time, rather than milliseconds in terms of thousandths of a second, now we're even up to a second, or if you want to consider it in milliseconds, could be as long as 300 milliseconds, or 0.3 seconds. So we're seeing a very prolonged contractile period here. Now some of the things are going to be similar, right? So you see depolarization due to sodium influx. We see repolarization due to potassium efflux. But we see this broad plateau here due to calcium. So that's an important distinction or comparison there. And there are others to be sure, but this is a change in membrane potential of an autorhythmic cell. So again, we see time here, and we see membrane potential on the y-axis, and here we see something unique as well. So here we don't see any resting potential like we saw in a neuron or a skeletal muscle cell or a contractile cell. It's always changing. Never is it just sitting there flat. We also notice variable roles of, again, sodium and calcium and potassium. Not exactly the same as what we saw before. We see calcium entering the cell much earlier here compared to what we saw in the contractile cell. And that's very different than the role we saw for calcium back in the neuron and in the skeletal muscle cell, where it was acting as a second messenger instead. Here we see calcium changing the membrane potential, much like sodium and potassium. All right, so that's certainly something important to know. All right, so that scales us down to the cellular level. Okay, we kind of spiral down to that microscopic level, and then we, like usual, kind of come back out again and talk about the general function, how that scales up from there. So the electrocardiogram, or ECG, that you see at the top here looks somewhat similar to what the graphs we were seeing already, where we see millivolts or membrane or potential, not necessarily membrane potential, but potential on the y-axis and then time on the x-axis. But the ECG is actually looking at the summary of the electrical activity of many, many cells, of the contractile cells that make up the heart, etc. So you definitely should know the electrocardiogram and what the electric events that we have going on there in terms of the P wave and the QRS wave complex, and then the T wave, what those correspond to in terms of what's physically happening with the heart. Now past that, that's looking at the overall electrical activity of the heart. The other ways that we can assess or estimate what's going on in the heart is to look at how much blood flow it's generating and things like that. So we start talking about things like cardiac cycle and what's happening when different chambers are contracting, what's happening when different chambers are relaxing, etc. We do some calculations in terms of how much blood might be exiting the heart at a particular time, which could have to do with how much blood is ejected in an individual contraction, but then how many contractions we have per unit time, and what things can affect that. And then from there, we move to the vascular part of cardiovascular. So with the vascular part of cardiovascular, you can see that there's a lot of different blood vessels. A lot of people just go, oh, yeah, I know arteries, and I know veins, so I'm good. But on the third level are capillaries, which are very critical blood vessels. But then you'll also notice that there are a variety of different types of arteries and veins. So we have elastic arteries and muscular arteries and arterioles, which are like small arteries, and venules, which are like small veins. Each of these different blood vessels are different anatomically and then functionally. And so you should be able to compare and contrast these different blood vessels, right? So we've got like 6 different blood vessels if you count these smaller specific types. And so you should be able to compare and contrast those blood vessels in terms of their structure and in terms of their function, right? We don't have different types of blood vessels or different structures unless there's a different function that goes along with it. Okay, so at that point, it's kind of a tour going through these different blood vessels and talking about their role and talking about their structure. So we're talking about the arteries, the big pulsing blood vessels that are receiving these tidal waves of blood from the heart when the ventricles contract. So they're kind of under the most physical stress from the blood that's arriving to them. And we talk about the difference between elastic and muscular arteries, and as the name would indicate, there are some differences in terms of their elasticity or their relative strength or ability to contract. We talk a little bit about measuring blood pressure and different effects of blood pressure. Then we get to the arterioles, which are the small arteries, which have a critical function in terms of working to slow down the blood and gets rid of this pulsing that occurs up in the larger arteries, right? Because the capillaries, we're on our way down to the capillaries. The capillary beds are very critical. The capillary beds are the place where our different substances are going to exchange with the tissues, because that's the main role of this cardiovascular system, is the distribution of substances around the body and the exchange of those substances. So much like the U.S. Postal Service, you've got kind of long distance kind of deliveries that you make where you're not making stops, you're just really getting it from, say, one state to another. But when you slow down and you get into the individual neighborhoods, you need to be able to deliver these items to the places that need it and pick up from those places that have substances to get rid of. So that occurs in the capillary beds, and their structure reflects that. But before we get to those capillaries, which are actually very small and very delicate, we've got to make sure that this pulsing tidal wave of blood that we see in the arteries is calmed down and slowed down so that this delivery can take place in the capillaries and we don't blow them out. So that's the role, the generalized role of the arterioles. Now, one of the main ways that they do that is that they change their diameter. We talked about the size of the pipes and how much of an effect that has on flow and resistance. And so they can actively change their diameter like many other blood vessels by constricting or dilating the smooth muscle in their wall. And so that's the way that they can, like we said, slow it down, reduce the pulsing nature of the blood. There are going to be kind of long distance or extrinsic controls of that, but also local controls too. A lot of times when we're going through the cardiovascular system, you kind of focus on, oh, okay, I get it, the conduction system of the heart, that takes care of the normal heart rhythm. And maybe the brain oversees that like a manager and modifies it in some way, right, with like the sympathetic and the parasympathetic branches of the autonomic nervous system. But besides those overall systemic level effects and changes, there's also going to be local needs that may be served by dilating or constricting the blood vessels that go to or from that area, right? So if you think about like a mosquito bite or something like that, or a cut in one particular area, you're going to need more flow to that particular site, right? Whether it's to get rid of toxins, whether it's to deliver more white blood cells to the area to fight off an infection or whatever, right? So that individual area, like a centimeter square, is going to need more blood flow than maybe 2 inches away from that. So there's certainly a number of local controls or local factors that can affect blood flow and blood pressure to a particular area. So we kind of get into intrinsic or local and extrinsic controls. Okay, and then we get down to the capillaries. And even within the capillaries, the sites of exchange, we have different specific examples of types of capillaries. Again, structure and function relationship. But when we get to the capillaries, one of the things that we have to figure out is how do the capillaries do their job? We say that the capillaries are a bit like the Postal Service. They not only need to deliver to the tissues, but they need to pick up from the tissues. And so we go back to some of the different types of transport that we learned previously, like diffusion and facilitated diffusion and even osmosis and things like that. And we need to understand how those different modes of transport allow the movement of these critical life-sustaining substances at the capillary beds. So you need to know different types of transport, different molecules that are transported in that way, etc. So a lot of it's diffusion. Like we said, there's osmosis involved as well. And bulk flow, or the exchange of fluids with the tissues, is important too. A lot of times we think, oh, well, why do we need to deliver fluid to the tissue? The tissue's already there. The cells are already there. They don't need to drink or anything. But, you know, if you really think about it, if you get dehydrated, then all those tissues in your body can also become dehydrated. They can lose intracellular fluid. They can lose extracellular fluid, etc. And in order to get that fluid back to them, first you need to drink or retain water, and then that newly found water, if you will, needs to get distributed to the cells that need it. So we talk a bit about bulk flow and how water moves into and out of the tissues as needed. Then we wrap around the other side and we go to the venules and the veins. Venules and the veins that are fairly simple compared to some of the things that we were learning before about capillaries or arterioles or different types of arteries. So as we kind of get around to the end of the macroscopic parts of the cardiovascular system, the heart and the blood vessels, we talk a bit about how the system is controlled, how it integrates with other systems, how the nervous system works to coordinate it, etc. And then, after going through some examples of when the cardiovascular system function goes awry, we start talking about the blood as well. So when you get to blood, the blood overall is the simpler of the three parts. But when we look at the blood, we also want to separate it out, piecemeal our knowledge of it so we can build into it and realize that there are going to be what we call the cellular elements, which include the red blood cells, 5 different types of white blood cells, and then platelets. So we have the cellular elements, but then we have the fluid component, right, the plasma. And again, if we look at the cellular elements, we have many different types of cells. We wouldn't have different types of cells unless they have different functions. So you need to know the functions of the different cellular elements. But then when people look at the plasma or think about the plasma, they think, oh, well, that's just a fluid to get the cells around. It doesn't have any real job. But you need to know that there are numerous ions and molecules important to the body that are contained specifically within the plasma. So because they're located within the plasma, that gives the plasma that function for the body, like transporting carbon dioxide or hormones for the endocrine system or something like that. So you need to know the different substances that are transported in the plasma and their particular role in the body. So don't forget about the plasma. Okay, so we talk about their particular functions, different types of cells and their particular functions, how they are birthed or how they are created, how we get rid of them. Some, again, cases, kind of pathogenic, or not pathogenic, but pathophysiological cases where there are diseases or disorders associated with the blood. Talking about platelets. And when we get into platelets, or these cell fragments called thrombocytes, we get into one of a few different physiological processes associated with the blood, part of the cardiovascular system. And those are things like the clotting cascade and hemostasis. And so we know that this is a closed circulatory system. The blood is supposed to stay in these blood vessels. And so what happens when you get something that creates a hole in a blood vessel, or you start leaking this precious fluid into the tissues or out of your body, etc.? What are the steps that are involved in order to get that to stop? And that's certainly an interesting process. It's also one of our examples, one of our few examples in the body of a positive feedback system where an initial change, like the formation of a blood clot, actually stimulates more change and amplifies that change, where we make more and more blood clots and we seal off this blood vessel that may be damaged and try to repair it. So you need to know the different steps of hemostasis as well as how we form a blood clot. Now you don't need to know all of the individual plasma proteins that are involved, but when you get down to some of the key points where we're looking at things like thrombin and fibrinogen and plasmin and things like that, those are some of the very common to these two different pathways, intrinsic and extrinsic, that we'll learn about. Those are the common parts of the pathways, and those are the final steps that are so very important in forming a blood clot, where we can seal off this broken blood vessel and give it time to repair itself. Okay, so you need to know about the clotting cascade as well. Now, like I said, you don't need to know, that factor 12 activates 11, activates 9, activates 8, etc. The most important parts of these is, for instance, that you know that there are two different pathways, how are they different in general, and then ultimately those last few activation steps, prothrombin to thrombin, fibrinogen to activated fibrin, etc. This is also a key aspect that you need to know. How do we get rid of one of these clots? We said it's a positive feedback system. It should just continually form a clot, a larger and larger clot. Why does that not happen? So knowing the activation of plasmin from plasminogen is certainly an important aspect about this physiological process too. And then towards the end of the blood section, we have, again, different chemicals, etc., conditions that are closely associated with this topic, this third topic of the cardiovascular system. So see, that wasn't so horrible in terms of cardiovascular system, if only, the outline was what you totally needed to know about it, right? But, like I said, take it in a stepwise process. You got 3 different significant parts for cardiovascular system, so take a step at each of those. Focus on one of those at a time and break that chapter up so that it's not something that's quite so overwhelming that way. Remember that you're going to be listening to the main lectures that we have, the archived lectures, etc., but there are oftentimes a number of videos, animations that I have loaded in the content area of Desire to Learn. And so different chapters will have different animations on different topics and different numbers of animations, but usually they're quite beneficial, especially for systems like this, where it's a very active system. We have movements going through, and it's hard to sometimes picture how it works in a piecemeal way when you're just seeing one slide and then one slide and one slide. So questions about things that we touched on tonight? Have people started the cardiovascular system? Why don't you give me a yes or no on that? Vanessa has. Ted, Monique, Megan, Mariello, Lakewood, David. How about you guys? Not quite yet? Not quite yet. So Vanessa's a little bit ahead of the game. Well, as David was talking about, it is a big chapter. It's a complex system, so you want to allocate your time accordingly. I realize you're probably waiting a little bit, too, to kind of have the discussion section occur so you can see the high points of it. But you want to make sure that you move into it in short order. With just a week out, and as David was saying again, kind of complex systems included in this one, we're going to need to pace it out that way and spend time being able to learn it in a stepwise process. Like I said, even though we have 4 different chapters included on Exam 3, it's really those 3 major systems where the bulk of our questions are going to come from, right? Muscular system, endocrine system, and cardiovascular system. And if you're kind of weighing it out, even so, cardiovascular system is larger than those other 2. So you might see more questions having to do with cardiovascular system too. So questions on cardiovascular or any of the other systems that you're going through? I do have endocrine system lectures uploaded already. Everybody's all right for now? All right. Like I said, I will be sending out a reminder email with some of those details we talked about at the beginning of this session, leading up to exam number 3 occurring on Thursday of next week at 6 p.m. on campus. We'll also have our second quiz 6, that will cover the cardiovascular system by itself. That will be due Tuesday night at midnight, but it should be available this weekend to help you prepare. And then we also have the possibility of meeting again, maybe for an extra review or last-minute questions or whatever on Tuesday night of next week, if people would like to do that. So you can let me know if that seems interesting to you. I'll also see if there are any other documents or sheets that may be helpful. I think I have an extra kind of exam objective sheet up on Desire to Learn already. We'll be using those chapter review questions too as a good guide point to important topics in the chapter. And any other things that I can think of, I'll let you know about and post up there too. Alrighty, well, I'll hang out for a few minutes like usual in case questions come up. Otherwise, we will be in communication through Desire to Learn. No fun this weekend. You stay home and study now. Alright, email me with anything you need.